So I'm Mary Kuhner of the University of Washington. I'm going to talk about three problems that sound completely unrelated at first, but the theory and the mathematics behind them turns out to be really similar. They're all questions of what can we find out about the past of a population by looking at the genes that it's got today. And the three questions are, where did the tragic HIV epidemic in a children's hospital in Libya come from? How many gray whales were there before we started hunting them? And how come some people with the condition Barrett's esophagus get throat cancer and others with the same condition do not? Modern genetic analysis can tie these three questions together. The tools that we're using to examine them are surprisingly similar, even though the questions at first glance seem really diverse. What I'm going to ask is, the talk falls into three parts. If I say something that doesn't make sense, please interrupt me with questions whenever you need to. If you want to ask more general questions about the topic, there's a question slide at the end of each of the three parts. So let's save the general questions for the end of each section. So, in 1998, some children were found infected with HIV in Libya. This may not sound surprising because we know there's a serious epidemic in Africa, but generally when children get AIDS, they catch it from an infected parent at or around the time of birth. These were older children. There was no obvious reason why they should have been coming down with HIV. People looked around, and more infected children were found, for a total of over 400 infected children in that area, which is a major tragedy. All of them turned out to have been treated at the same hospital, El Fatih Children's Hospital in Benghazi. All of them had had surgery or some other kind of invasive procedure there. 43% of these children also had hepatitis C, so it's a double whammy. This cries out for some kind of explanation, and there are three possible theories. It could be some kind of coincidence where the children gained these infections in their community, and they just happened to all have visited this hospital. The children could have been infected at El Fatih due to some kind of poor sanitary practices, that is, by accident, by some kind of medical mistake that allowed the virus to spread. Or they could have been infected at El Fatih by somebody's deliberate action infecting them, a murder theory. The Libyan government seized immediately on the murder theory. It was the most acceptable to the parents and relatives of these children. They arrested 5 foreign-born nurses and a foreign-born medical intern who were working at El Fatih. Libya then reported that the accused people had confessed to deliberately infecting these children and sentenced them to death. The scientific community was really skeptical about these alleged confessions, which no one outside Libya had access to. And they wondered why anyone would have been motivated to do such a thing and to try to carry out such a large and difficult to hide conspiracy. So there was interest in whether genetic evidence could help establish whether these allegations could possibly be true. The alternative of going to El Fatih and asking questions was very difficult to do because of the political situation in Libya. So how can we do anything with genetics here? HIV and hepatitis C virus, like many viruses, mutate very quickly, so quickly that no two infected people have exactly the same virus. It changes very, very fast over time and in the transmission from one individual to another. And you can see relationships among viruses that have the same source. An early use of this was made in the 1990s. Patients of a particular dentist were found to be infected with HIV. The question was whether they'd been infected somehow by their dentist or whether they'd picked up those infections in their community. And the question was settled by discovering that the virus in these patients of this dentist were very closely related, much more closely related to each other than to viruses found in the community that they came from, suggesting very strongly that they had a common origin. And the virus that the dentist himself carried was a member of that tightly clustered group. So there's information here we might be able to use. How can you infer a relationship among viruses? This is the most technical slide in the talk, but I'll go through it quickly. These are hypothetical DNA sequences. Whoa, I can't point with that. These are hypothetical DNA sequences from multiple individuals, so individual 1, 2, 3, 4, where the individuals here would be virus sequences. In many positions, they are all the same. That's how we know it's the same gene in the virus and can line them up and correlate them. But there are differences due to genetic mutations. For example, these 3 sequences share a C where every other sequence shares an A. The simplest explanation of that is that those 3 sequences go back to a common ancestor in which a mutation from A to C occurred, or perhaps all the other sequences go back to a common ancestor in which a mutation from C to A occurred. We don't know off the top which of those it is, but it suggests a clustering of these 3 sequences. Similarly, this T as opposed to C suggests a clustering of the sequences with T together and with C together. You could represent these clusterings by drawing an evolutionary tree showing how the sequences are related. One possible evolutionary tree is shown on this side. It says, for example, that these 2 sequences are closely related and they're part of a larger group that also includes these up here. The mutation to that T could have occurred on this branch and been inherited by all of the descendant sequences. You'll notice that there's more information in that evolutionary tree than these sequences really support. Some of the branches don't show any diagnostic mutations, so there are other trees that would fit this data equally well. That's often going to be the case in population analysis because the number of mutations separating, say, HIV viruses from people who got their infection in quick succession, it isn't very large. And so a lot of the mathematics that underlies the work I'm going to talk about has to do with dealing with the uncertainty. The fact that this tree is plausible, but other trees are also plausible. You could rearrange some of these branches that don't have any mutations on them and get something that fits the data equally well. And so a lot of the mathematics will involve taking sums over many possible trees that could explain the data. So what could this kind of evolutionary tree tell us about the guilt or innocence of the foreign-born medics? Well, to start with, if children obtained their infections randomly in their community, and it was just a coincidence that they'd all been patients at El Fatih, we would expect the children from the hospital to be scattered amongst samples of other viruses taken from their same community. Wouldn't expect them to cluster together if they obtained their infections independently from different sources. Whereas if they all got the infection from a common source, either bad sanitary practices at the hospital or deliberate infection, we'd expect those sequences to cluster together, to be more closely related to each other than they are to other sequences found in the population at large. An international team was very quickly pulled together to obtain DNA samples from the blood of 44 of the children who'd been brought to Europe for attempted treatment and study. And they compared them with the worldwide database of HIV samples, specifically noting African samples from Egypt, Cameroon, and Ghana. This is my acquaintance Tulia de Oliveira, who was the first author on the resulting paper. They were helped by Lawyers Without Borders, which is an association like Doctors Without Borders that tries to provide emergency legal assistance in situations like this. I want to tell an anecdote about this paper. Several months after it was published, I got to speak with Oliver Pybus, the second author, who's an acquaintance of mine. I've always thought of Oliver as a pretty happy-go-lucky guy. And I asked him what it had been like to publish this rather high-impact paper. He put his head on the table and said, "Mary, I thought I knew about deadlines. I'd had grant deadlines where I might not get the money. I'd had paper deadlines where the paper might not get published. I'd had exam deadlines, but I had never had a publication deadline where 8 people might die if I didn't turn in my paper on time. And 4 months later, I'm still shaking." So it's a situation scientists don't necessarily find themselves in every day. Just to quickly refresh those of us whose African geography is a little shaky, here's the relevant part of Libya, Benghazi in Libya. Here's Egypt, and then Ghana and Cameroon, which is in an area that's somewhat the heart of the HIV epidemic in Africa. So I apologize for this slide. The journal was happy to let me have the slide, but not happy to let me have a high-resolution version for some reason. But this is an evolutionary tree of HIV sequences from around the world. This large triangle is the sequences from the 44 children. The relationships amongst the sequences infecting the children can't be distinguished very well, but they're all really, really similar to each other. Much more similar to each other. Note that long branch separating them from the rest of the tree. They're most similar to a sequence from Ghana, but not particularly similar. So it looks very much like, as far as HIV is concerned, all 44 of the examined children probably were infected from a recent single source. Hepatitis C virus comes in several subtypes, more than one of which were found in these children. For subtype 1, we get a very different picture. There were 2 clusters of related viruses in the children, plus some strays here and here. They were related to various sequences, many of them from Africa. This looks more like the picture you would get if you just picked random cases of hepatitis C from the community. They're mixed in with other sequences from the community. They're not forming a single tight cluster in the same way. This is the worldwide epidemic strain of hepatitis C virus, and it may be that some of these children contracted it in a way that was not related to whatever single catastrophe led to the HIV infections. Hepatitis C type 4, some of the children, a smaller number, were infected with that. All of those infections look like they have a recent common ancestor. Again, they form a cluster to the exclusion of everything else in the tree. This strain of hepatitis C has an interesting and somewhat tragic history. It's not common worldwide, but it's extraordinarily common in Egypt, because in the 1970s, some anti-worm medication broadly distributed across Egypt was unbeknownst to anyone infected with hepatitis C. So that set off a local epidemic. But there are Egyptian sequences in this tree, and this is not a random sample from Egypt. This is a single origin again. So for 2 of the 3 viruses being looked at here, the genetic evidence, the first pass on it, says these children were infected essentially from a single source, either from each other or from some common source in the fairly recent past. This doesn't look good for the medics. It certainly seems to rule out, for those 2 viruses, the community theory that says that the children didn't get these infections at alpha T. It's pretty clear at this point that they did. Maybe not hepatitis C type 1. Those may have been accidental addition to the story, but the other 2 really look clustered. But that doesn't answer the legal question, was it accident or was it murder? We know that the medics arrived in Libya in March of 1998. When was this common origin of those strains becomes the critical question. If the common origin was after March 1998, that could represent the medics coming in with a bottle of contraband, say infected blood, and infecting children starting on that date. But if the common origin of these infections was well before March 1998, that theory won't really fly. It's very difficult to imagine that someone who was setting out to infect children would bother to get a whole bunch of different but closely related strains, put them in separate bottles, and infect a child with each one. If the common origin of the infections that hit these children is prior to the arrival of the medics in Libya, it's almost sure that the medics couldn't have been involved in doing it. How can we date this kind of thing? Well, there's a complication. Libya challenged our ability to do this at all because they said you're going to have to use estimates of how fast HIV mutates in the wild. We don't think this strain came from the wild. We think it's a genetically engineered terrorism weapon. Some evidence bearing on that could be found when we sequenced, I shouldn't say we, I wasn't personally involved with this, but when the virus was sequenced. It's a member of an HIV subtype, CRF02AG. It's a recombinant in the fairly distant past between subtypes A and G. It's very prevalent in Africa. So it's not a novel type of virus that had not been seen before. And you can ask, how far has this virus diverged from the common ancestor of all the CRF2 AGs? So we compared it to other AGs in the database. It's very typical. It's the common ancestor of that strain was a certain number of years ago. I don't have it memorized, but maybe 40 years ago. And it has about as many mutations separating it from that common ancestor as any of the strains found in the wild. So there's nothing about the genetic sequence of it that makes us think it's some new thing whipped up in the lab. It looks like an HIV virus that you might very reasonably encounter in the wild in Africa. And therefore it was considered reasonable to assume that it's probably got the mutation rate of HIV viruses encountered in the wild. So my colleagues used a software package called BEAST to estimate how many mutations back to the common ancestor and to convert that to years, knowing worldwide information about how rapidly HIV mutates in patients. The critical thing that they use the sophisticated software for, you could get an estimate of the time just by taking the trees I showed earlier, measuring the number of mutations back to the inferred common ancestor, and applying the mutation rate to get a time. The problem with that is it doesn't give you error bars, and error bars are critical here. Suppose that we found the date is before March 1998, but it's got an error bar of plus or minus 4 years. That's not going to exonerate anyone. On the other hand, if there's an error bar of plus or minus a month, we might be all right. So the statistical power was plowed into looking at many different hypotheses for how the children are related, looking at a range of values for the possible mutation rates, so as to get an accurate error bar to tell us whether we've really got exonerating evidence or not. And the result, perhaps to no one's surprise, but to a lot of people's vast relief, is that the evidence is very strongly against the virus having been recent. Here is hepatitis C type 1, hepatitis C type 2, type 3, and HIV. And in all cases, the error bars exclude an origin on or after March 1998. So it looks like while these viruses have a common ancestor, that common ancestor was probably already present in El Fatih Hospital before the foreign medics even arrived there. And therefore, they cannot be responsible for bringing it in. This paper was published, and the medics were extradited to Bulgaria by the Libyan government. The Bulgarian government then pardoned and released them. There was some international kerfluffle about whether they had the right to do that, but there wasn't much Libya could do. In August 2007, the genetic analysis was vindicated by Saif al-Islam Gaddafi of the dictator Gaddafi's family, who confirmed that Libyan records showed that some of the children had in fact been known to be infected prior to February 1998. That is, they were known to have been infected before the medics arrived in the country. He also talked about the confessions and confirmed that the confessions were obtained by a mixture of torture and threats to engage in terrorist activity against the medics' families if they didn't confess. I'd like to say something here. One can have various feelings about the morality of torture, but I'm here to say as a scientist also, it doesn't work. You can torture people and they will answer your questions and they will tell you what they think you want to hear, but as a means for finding out the truth, it's useless, and I think this shows it. I think it's very likely that the medics did in fact confess to anything they were told to confess to, but it was useless as a means of finding out what had actually happened to the children. And the tragic thing is, while the medics were in jail, they were in jail for 9 years and 8 years, I think, in Libya. It's not totally clear that whatever was going wrong at El Fatih was being addressed. So they were a distraction from finding out what the real problem was. Very likely, international sources think, it was inadequate sterile practices and reuse of equipment. This is a huge problem. Anywhere that's under economic stress, it's very tempting to reuse hospital equipment that in the US would be single-use only. But unless you're scrupulously, exactingly careful about sterilization, it's very, very unsafe to do so. So, subsequent to that, Libya officially acknowledged that while there was negligence, there was a disaster that took place, there was a tragedy, and this is all true, but it was not a deliberate act. So, the story has a happy ending for the medics. It does not, alas, and is not going to have a happy ending for the children. Childhood HIV infection in Africa is close to a death sentence. We're fairly good at treating adults in first world countries. We're not nearly so good at treating children, and the situation in Libya is so unstable that the consistent lifelong treatment needed is sadly improbable. About 40 of them have died so far, but the prognosis for the rest is not good. So, questions on that part of the talk? Yeah.
Would there have been a general outbreak, not just among children? So you'd have 400 children, but say 1,000 other people who also contracted HIV at the same time?
Well, the question is why there wasn't a more generalized outbreak, and I think the answer is that El Fatih is specifically a children's hospital. It doesn't treat any adults. And so you get this tight clustering of pediatric cases only, which is on a piece of corroborating evidence saying that the infections arose there. There's a suggestion in Wikipedia that a patient 0, a child who was admitted over and over during this period, who was perhaps infected and was then the source of these infections. I haven't been able to see the primary evidence for that, so at this point I'll just put it forth as a hypothesis. But we know that HIV is a very short-lived virus outside the body, so it is startling that you could get this kind of over time consistent infection. But if you're continually readmitting the patient who's the source of these infections, then you've got this opportunity to continually infect new children from that same single source. And that's a terrible mistake. I don't think anyone's specifically to blame other than the general practices of the hospital were not adequate to contain infections. Other questions? All right, going to turn to what I find a more cheerful topic, which is the gray whale, a fascinating, magnificent animal of the ocean. The gray whale is currently protected. People are trying to rebuild its population. We'd like to know how many whales were there before whaling, because that's in some sense the target that we might want to aim at for assessing population recovery. It's difficult to manage conservation of an organism unless you know about how many the ecosystem can support. You might be shooting for a number that's too high or too low otherwise. The research I'm going to talk about here, I was a little bit more directly involved in. It was done by Elizabeth Alter and Steve Palumbi, and then Eric Rines, who was my research technician at the time. His name is on the paper rather than mine because he did all the heavy lifting, but the software used was software developed by my lab. So the question is, how many gray whales were there pre-whaling? We know at the end of whaling, the population was in desperate trouble. It's recovered to some degree since then. Whaling ship records have been the normal way of trying to find that out, but they're not conclusive for a number of reasons. Whalers worried about what we'd now think of as commercial espionage, other people finding out where the good whaling beds are, and so there's some evidence that they may have sometimes falsified their records to conceal large numbers of whales that they found. And also the formula for going for how many whales did a whaling ship see to how many whales are in the ocean is itself controversial. The current status of this animal is that there's a very, very small amount of legal whaling, mainly on behalf of First Nations peoples, particularly in Russia, about 140 animals a year. There's also some poaching, especially in Asia. One of the reasons I really like co-author Steve Palumbi is he did a beautiful piece of espionage where he pocketed a DNA testing kit and went on vacation to Japan and bought legal whale meat in supermarkets across Japan and ran it through his little pocket testing kit and found that quite a bit of it was not what it was supposed to be, and a certain percentage was species that it is not legal for the Japanese to hunt. There was a big fluff, understandably, about this, and I hope that it's led to some tightening up by the Japanese government on what they're allowing to be whaled. After whaling was essentially terminated, gray whale populations began to recover, but lately that recovery seems to be slowing. And the question is that because we've gotten back up to pre-whaling levels of the animals? Or are the populations slowing because they're being suppressed? Could be by there's still too much whaling for their good health. Could be that there are environmental pressures that are preventing them from making a full recovery. We'd like to know what's going on. Whalers' logbooks are not giving us, this is an actual photograph of a whaler's logbook where the whaler not only counted but illustrated the animals he found, but they are not giving us really what we want to know. Molecular data gives us an alternative possibility for finding out what the past history of this population was. So, Eric and his working group picked out genes that could be sequenced. How do you get genes out of a whale? It's fairly easy to get a very small blood sample from a whale without doing the whale any harm. And this was actually, I think, piggybacked onto tagging attempts. So you get a tiny amount of blood, you do DNA sequencing. They looked at 10 genes, 7 ordinary genes from the paired chromosomes that whales like humans have, 2 sex-linked genes, which tell us something a little bit about the sex distribution of the population, and then the mitochondrial DNA, which is passed only from mother to offspring in whales as in humans, and tells us something particularly about the female population size. There were a number of issues that complicated this study. Different genes may have different mutation rates, and that's going to have to be calibrated. These genes exist in different numbers per individual. I have 2 copies of most of my genes. I'm a female, so I also have 2 copies of my X-linked genes, but the males in the audience have only 1 copy of each of those. So the total number of copies floating around in the population is a little lower. And then the mitochondrial genes, I essentially transmit only one copy to my offspring, and males don't transmit any copies at all. So the population size is effectively quite a bit smaller for those genes. And those things have to be calibrated. A trickier one is that genetic methods can only measure the breeding population. We're going to look at the web of relationships among individuals tracing back to common ancestors. Individuals that never had any surviving offspring are essentially invisible in that analysis. So the population of successful breeders is always, in all species, smaller than the number of heads swimming around in the ocean. The trick is to find out how much smaller. And then finally, there's some population structure. Gray whale populations are not one big sloshing gene pool throughout the entire world's ocean. They actually stay in relatively discrete areas. There's some mixing of the populations, but not complete mixing. So Elizabeth and her team worked on all of these factors, trying to make conservative. It's important to be conservative here. Because about halfway through this project, we got a phone call from the California State Legislature saying, could you please testify on the endangered species status of gray whales? So this is not just an academic question. This was going to drive California's decision about how to manage their whale populations. Go see that. So the question that leaps to mind is, how can modern data from a relatively smaller population reveal past population size? How can population data reveal population size in general? How does that work? Well, the bigger a population is, the more distantly related a random draw of two people or whales from it are going to be. If you imagine a little village where everybody's everybody else's cousin, and they've been living there for thousands of years, and it's just this little isolated gene pool. If I pick two people from that population and look at a particular gene they have, the common ancestor is probably pretty recent, because the village is pretty small. There aren't that many genes to choose from. All these people are each other's fourth or fifth or sixth cousins. If I go to New York City and I pick two people at random from New York City and ask how long back in the past to the common ancestor of gene copies from those two people, the answer could be an awful lot longer. Could be hundreds of thousands of years. Could be millions of years. It depends on the gene. But in general, there's a relationship between the size of the population and the time back to the common ancestor of gene copies picked from that population. And that's the information we're going to leverage. The problem is, whatever we know about the whales in the past, we know that their population shrank dramatically as a result of whaling. In fact, if whaling hadn't stopped, I think that we would no longer have any gray whales at all. It was pretty touch and go for a while. So how can you find out about the past size? Hasn't all of that information been destroyed, essentially, by whaling? And to understand why that's not the case, you need to know a little bit about what happens when a population shrinks. And I have a picture here, which I think is more helpful than 1,000 words. So this bubblegum machine is a gene pool, and the different colors represent different genetic types, different alleles at the genes in the population. A big population will be very diverse. It'll have a lot of different bits of genetic variation within it. Just as if you imagine just looking at a crowd of New Yorkers, you can see there's a lot of genetic variation present. In that small hypothetical village, if you look at the people, they're going to look much more similar. There'll be a lot less variability. What happens when you take a population like this and suddenly shrink it? The immediate thing that happens is you just have less gene copies, but much of the diversity is still present. If we boiled down the human species to a couple of hundred randomly picked New Yorkers, we'd still have a group of very diverse people. They wouldn't contain all the diversity of humanity. There's no room for it. But they'd still be quite different one from the next because they came from a big population. So immediately after the shrinkage happens, there's still quite a lot of very dissimilar genetic types in the population. As time passes, many of those will be lost if the population remains small. And you'll eventually get down to a much more homogeneous population where most of the genes trace back to recent common ancestors. But this takes a little while. And if you look at whale population sizes and whale generation times, which are long, they're comparable to humans, so it's only a few generations ago that whales stopped being hunted, in fact, you'll see that we would expect whales to still retain a lot of the genetic diversity that they came into the whaling era with. It has not had time to be completely lost. The great danger in conservation biology isn't the shrinkage so much as it's the persistent smallness going on for a while. If the population rebounds, it can actually retain a lot of its diversity. But the longer a population remains small, the more of its birthright of genetic diversity it's going to lose. We're not at that point yet with the gray whales. We would expect, a priori, just looking at the math, that they would still retain diverse genotypes from their ancestral population. The question now is, how diverse are they, which is a measure of how big was the ancestral population. We use software written in my lab, the Lamarck genealogy sampler. It looks at many, many hypotheses for how the sample of whales was related for each of these genes. It takes into account the population structure, the division between the Atlantic and Pacific, the North and South Atlantic, and gene flow among those populations, the number of copies of each gene in the population, which varies, as I mentioned, between regular genes, X-linked genes, mitochondrial genes. It uses mutation rates that were estimated based on fossil calibration. If we see that populations are diverse by a certain number of mutations, that doesn't quite translate into what we want to know unless we know something about how fast mutations accumulate. And the way that that was found was by looking at events in the whale fossil history where we have pretty good dates. For example, you can often date things because if two bodies of water were not in contact and then it came into contact, then any species found in both got there subsequent to the point where the bodies of water came into contact. And the result of this is estimated breeding population sizes with, again, error bars. Error bars are critical here also. I'm not going to be able to say to the California legislature, well, I think there should be 100,000 whales plus or minus 90,000. That's not going to be of any use to them in making policy. And if that's all the genetic data will tell me, I have to be scientifically honest and say the answer is we don't know how many whales there were, not in any useful level. Just a quick additional note on why the head count is different from the breeding population size. Well, if you go out and count whales, you're counting some individuals that are too old or too young to breed. They're not contributing to the current breeding population. You're counting some individuals that are fated to die young or fail to find mates. And also, not a great deal is known about gray whales' mating strategies. In many species, if you think of deer or cattle, a few males disproportionately monopolize the females. And that reduces the breeding population because the unlucky males, while recent research suggests there's more cheating in these species than we thought, they're at a reduced rate of reproduction compared to the successful males. If you want a really dramatic example of that, look at thoroughbred horses sometime and ask what proportion of current thoroughbreds are descended from Secretariat. It is astonishing how many of them are. And that's reduced the genetic effective population size of racehorses. If Secretariat, it was a magnificent animal, but if he had any genes that we didn't want, we've now got tens of thousands of copies of them. So studies of whales suggest that the breeding population is actually only about 1/6 of the head count population. So the numbers I'm going to show have been corrected for that to go from the genetic estimate of breeding population to an estimate of how many whales swimming around that should actually mean. So this slide is busy, but I'll go through it a little bit. 7 autosomal genes studied in 70-odd whales apiece. The estimates vary from gene to gene. There's some noise in them. X-linked genes, mitochondrial genes. This locus is really, really different from the others. The others all show fairly high estimates. This one's actually outside the error bars of the other. What I think we've hit here is probably natural selection. There was a genetically advantageous mutation at this locus, and whales carrying that mutation had a higher rate of survival, and so that particular genetic type has overpowered the others and is present in most animals. One of the reasons for having 10 genes is to try to avoid being swayed by that. If we'd seen only this gene, we'd end up with a very small estimate for how many whales there are. With 10 genes to look at, we can say this looks like an outlier. Perhaps something different is going on at that locus, and further study could be done there. The results I'm going to show, they actually ran the results with and without this locus. It actually does not change the conclusion very much. The evidence from the other loci outweighs it. So putting all the data together, they get an estimate of just under 100,000 individuals in the breeding gray whale population. That number doesn't probably mean much to you or me until you put it in context of what we had in 2006 was about 20,000 whales. And what we thought was the natural carrying size of this population pre-whaling, based on the whalers' logbooks, only about 20 to 30,000 whales. So the genetic evidence is saying there were something like 4 or 5 times more whales in the ocean pre-whaling than we had realized by examining whalers' records. I kind of suspect the whales had managed to find somewhere to go where the whalers were not finding them and not counting them. I don't know. It may also be the whalers lied about them. It may be our conversion factors is wrong, but these numbers are really discrepant. Here's the error bars on the overall estimate, 78 to 117,000. This number and this number right out, well outside the error bars for that. I wish, in the sense, that Steve Palumbi were here to talk about this, because he's a whale biologist and I'm not, and he can wax eloquent about what 100,000 whales mean to an ecosystem. This is an ecologically important animal because its feeding strategies involve resuspending sediment from the ocean floor, pushing it up into the sunlight zone, which increases the fertility of that area of the ocean. That doesn't sound like a big deal, but 100,000 whales is a lot of whales. Palumbi estimated that that would move 700 million cubic meters of sediment, which is about 12 times as much sediment as the Yukon River puts out. And if any of you are Alaskans, you know there's a huge fertile delta around where the Yukon reaches the sea. Palumbi estimates that just to start with, you'd have about 1 million more seabirds in that part of the world than you do now because of the increase in ocean fertility driven by these animals. His practical conclusion from this, I'm a population geneticist, not a conservation biologist, so I can't contribute to that part of the discussion. But he suggests that we're killing about twice as many gray whales a year as we can afford to, that we should back off at least that far. I personally would love to back off all the way if possible, and see if we can get the population to recover to the actual historical level with the concomitant, probably, improvement in the northern Pacific ecosystem. Palumbi's repeated this type of analysis with minke, humpback, and fin whales. The interesting thing about it is that humpback and fin were preferred whales for whalers. And in both cases, we see the same big discrepancy between the logbook entries and what the genetics says about the number of animals. Minke whales were not preferred. They were considered kind of a trash whale that you would catch if nothing else was available. Their population didn't come down as far. It's recovered pretty nicely, and there's less of a discrepancy. The numbers it's recovered to look like they may actually have been pretty close to the pre-whaling historical level. But that's not at all true for humpback or fin, as it's not true for gray. So the great contribution of genetic data here is that populations of marine animals in the present are hard to count. Many of the users of my software are marine biologists, because the numbers and distribution of a lot of marine animals are extremely cryptic. A student told me a wonderful story, which I've cherished ever since. She worked on evolutionary history of beaked whales. And there's at least one species of beaked whale out there that is known only from a transient photograph snapped by a National Geographic photographer from a ship. He looked out, he said, "My gosh, what is that?" and took a photograph. It's never been seen before. It's never been seen since. It's a very clear photograph, and she was confident that it's an undescribed species. And that tells you something about how big and how still mysterious the oceans really are. That something as large as a beaked whale, which is not a big whale, but it's a whale, can be out there. There are also 3 more species that we know only from 2 or 3 animals that have washed up on beaches over the historical past. So you see an animal in Australia and you see another one in Asia, 20 years apart. There must be a population, but where are they? The truth of the beaked whale is that the animal hates boats. And it's going to be a long time before we know very much about it. But marine animals in general are difficult to count. Marine animals in the past are practically impossible to count. And if you want to make an intelligent effort to conserve them, it really helps to know what you're shooting for. And it's really helpful to have solid scientific evidence, because one thing we know is that if the scientists are not sure, the political forces will put enormous pressure towards whatever answer is economically profitable or politically convenient. And the only way to stand up to that pressure is to have rock-solid science as much as possible. So there's 2 unanswered questions about these whales. The troubling one to me is, can we actually reach that number? There was an ecosystem pre-whaling that apparently could support about 100,000 gray whales in the Pacific. Does that ecosystem exist? One sad possibility for why the restoration efforts seem to be plateauing is that that ecosystem simply isn't available anymore, and that we're going to have to start doing a repair work from the ground up, or from the sea bottom up perhaps, in order to have a chance of ever getting back there. And then the other question is politically, whose problem is this? Whaling is difficult because the animals live outside the 200-mile zone. They're in open ocean. Every country has an opinion. The World Whaling Commission has an opinion. Poachers certainly have an opinion. Whose problem is it if too many whales are being killed to allow the population to rebound? The California legislature, I think I respect the fact that they want to make it their problem, but do they have the power to do anything about it? It's not clear. Well, I'm sure they could contribute negatively, but the question is, who's in a position to contribute positively? All right, so questions about whales? Yeah?
Not exactly about whales, but you said a little earlier that, and I think you were saying that a same gene in two individuals could have an ancestor, a most recent common ancestor, 100,000 million years past, which immediately struck me as unintuitive if the most recent common ancestor of individuals is clearly much more recent than that. But you were talking about genes, and is there any clarification I should be understanding about this?
So the question is, if people have common ancestors often only a few hundred or thousand years ago, how can there be some genes where our common ancestor is hundreds of thousands or millions of years ago? And the tricky thing is, we don't have just one common ancestor. We have a bunch, because the different parts of the different chromosomes and different parts of chromosomes reshuffle. And the thing I did my thesis work on that really drives this home is immune system genes. I might have the same immune system genes as, my great aunt, but I had quite a few ancestors back that many generations. I might have gotten my immune system genes for each one of them. And each of them had quite a few ancestors, and they might have gotten their immune system genes from each one of them. There's a statistical sorting process that if the gene variation isn't particularly important, tends to funnel you back to a common ancestor for the gene a certain number of generations ago. It's about 4 times the population size. But if natural selection intervenes, say attempting to keep more diversity in the population because diversity in that gene is survival important, then the common ancestor can in fact be much longer ago. My thesis work was on certain genes of the immune system where it's a huge survival advantage to be different from your neighbors. A population that's all the same gets nuked by plagues because as soon as one individual catches it, everyone around them is vulnerable. And people see this in, for example, inbred mouse strains, which are really notoriously hard to keep alive. The common ancestor of one of those immune system genes from, say, you and me may have existed in a chimpanzee. That's how far back those lineages go. So there's just variation across the genome. Some parts of the genome have recent common ancestors. Just about every single sub-Saharan African has an ancestor within a few thousand, common ancestor within a few thousand years at a particular malaria resistance gene. So that's really, really, really recent. And humans' common ancestors for immune system genes can really go back over 6 million years. In between is the massive genes where there's neither pressure for everyone to be the same nor pressure for everyone to be different. And on average, the common ancestor is about 4 times the population size. That sounds huge, but the human population size has not typically been 7 billion. It's typically maybe been in the range of 500,000. And so not extraordinarily long ago. I think there was some, yeah.
Do you know what the current acceptable whaling rates are for you?
What I read in Wikipedia, the question is, what's the current acceptable whaling rate? For gray whales, what I read is what's currently being taken is about 140 animals a year. Mainly First Nations in Russia that feel they have an ancestral right to whale these animals. There's no commercial whaling of gray whales legally at this time.
I had a different question. Is there a way in which a higher
The question is, whoa, oh dear. Charles, I've got water on your computer. You might want to blot it off real quick. I'm going to step away from this thing before I do any more harm. That better be my last question on this one. And the question is, how can we account for possible complications in the model? At least that's my best summary of that question. To get these numbers, they took a range of possible values for a lot of things, such as how much gene flow there was, how isolated the populations were. So the error bars are taking a certain amount of uncertainty in the model into account. And that's really the best answer I can give. We can do better currently than this study if someone can get sufficient whale data. People are going up to looking at very large chunks of the genome for this sort of relationships. And there's more information to be found. I don't think it's going to challenge the basic results. All right, I'm sorry. I know there are more questions, but I want to get my third bit in. So I'm going to stretch the definition of population. I've talked about populations of viruses. I've talked about populations of whales. I'm not going to talk about a population that exists within a single human body, namely a population of cells in the esophagus. Some proportion of people, particularly older males, have damage to the lower esophagus by gastric acid and juices, and develops an abnormal layer of tissue. 5% of these people then go on to develop a severe form of esophageal cancer, whereas the other 95% don't. The condition is called Barrett's esophagus. It's often referred to as a pre-cancer condition, but I think that is unnecessarily terrifying to people, 95% of whom are not, in fact, going to get cancer from it in their lifetime. Normally, the lining of the esophagus is a smooth, flat layer. The esophagus has to deal with food, but it's not really expecting to deal with concentrated hydrochloric acid. If something has gone wrong in the body and acid and other gastric juices are coming back up into the esophagus, some people's bodies react by changing the type of tissue that's lining the esophagus from the smooth, flat layer of cells into a structure that has these pockets called crypts. And this is a structure that's very typical of the lining of the intestine, which is a tissue that's exposed to a lot of acid and bile and so forth. So it's interesting. This is not a disorganized tissue. It's very carefully and logically organized, but it's organized differently than a normal esophagus. The cells have changed their fate from a normal esophagus lining to something that's surprisingly close to intestinal lining in the throat. Why would your body do that? That is really weird. It may actually be an attempt for the body to deal with this ongoing acid damage. The stomach juices are mutagens. They damage DNA. And that's a risk situation for cancer. The structure of these little crypts is that at the bottom of each one are stem cells. They divide and produce new stem cells, which remain at the bottom. They also divide and produce differentiating cells, which move up the sides of the crypt as they mature, and eventually are sloughed off into the esophagus. So a mutation down here, can have several offspring down here. It can produce cells that go up here, but those cells are going to die. There's no very easy way for a mutation that occurs here to spread across the esophagus. This is like little isolation cells. So each set of cells in its own crypt, producing its own offspring, and if one of them goes wrong in some modest way, it doesn't have much opportunity to spread. So there's some mysteries about Barrett's esophagus. Most people with Barrett's esophagus never get cancer. We follow them for decades and decades until they die of something else. But those that do get cancer often get cancer very quickly, usually within about 4 years of the detection of Barrett's esophagus. The clinical standard is to survey these people every 6 to 12 months, and very, very frequently, among the 5% that get cancer, they get it quickly. The other mystery about this is that the rate of this cancer is climbing rapidly in the Western world. It's the fastest growing cancer. It's a rare cancer. We're not in enormous danger from it. We want to know why it's growing. People suggested it was part of the so-called obesity epidemic, but it started climbing before obesity rates did. So something is going on there that we don't understand. So Rumen Kostadinov of Johns Hopkins and I became interested in how a tissue that's divided into crypts like this is going to behave, and we simulated it in the computer. So we made a system within the computer with simulated crypts that arose, divided, died out, and had mutations. And we looked at a case where there was no natural selection favoring one cell over another. We just wanted to know what's the natural dynamics of a tissue like this if no favorable mutations arise. So the interesting thing is we start out with an esophagus full of normal cells, divided into crypts, and then subject them to some rate of mutation and watch what happens over 20, 30, 40 simulated years. Nothing happens. Cells develop mutations, but the crypt structure does its job, and those mutations really don't spread very well at all. You get tiny, tiny patches of mutant cells. The story is very different, though, if you start the way that we think Barrett's esophagus actually starts, which is with a small number of cells at the stomach end of the esophagus, which spread upwards. So if a mutant cell happened to find itself on the leading edge of that spread in our simulations, then it did have a lot of potential offspring, and we get these striking patterns. This is a simulated esophagus. This is the stomach end, and this is the throat end. Imagine this picture wrapped into a tube, which is the shape the esophagus is. Down at the bottom, the cells are a gamish of cells, each which has a few individual mutations of its own, but they're very tiny patches, and so the color looks sort of brownish, green, and blurry. But as the cells colonize their way upwards, we get these large patches of cells that are very genetically distinct from each other. The colors here are essentially a measure of genetic difference. Each different colored patch is cells that contain quite a few mutations that are not found in any of the others. So we go from tiny patches carrying different mutations to big sectors all sharing the same set of mutations as the tissue spreads up the esophagus. And again, this is without any advantage or disadvantage to those mutations. That's just the neutral process taking place. We've seen this pattern before, not in esophagus, but it resembles a phenomenon that's been described in the last 10 years in population genetics called gene surfing. When a population is spreading rapidly across an area, mutations that are on the leading edge can surf to high frequency. This was actually first recognized in humans in the settlement of French-Canadian populations in Quebec. The French-Canadian population that immigrated to Quebec grew really, really fast. It's one of the highest growth rates humans have ever achieved. Average families, I think, around 12 to 14 children. And a genotype that found itself on the leading edge where there was new land, new fertile valley land to colonize, was rapidly increased. So you get these booms of random mutations that happen to be present in a few individuals. And just because that individual hit the jackpot of being on the leading edge of the expansion, now they have thousands of great-great-great-grandchildren. You can also see this in bacterial colonies on Petri plates. These are bacteria that have been genetically engineered with either a glowing red or glowing green protein. And then the two kinds have been mixed together, and a droplet was put in the middle of the plate. Initially, they're all mixed up. But as they grow outwards towards the edge of the plate, the only ones that can colonize all that vast new bacteria-free territory are the ones on the very outward edge, relatively small number of cells. And so one green cell on the very outward edge has a bunch of green offspring that grow up into these dramatic sectors, green offspring and red offspring, sorting themselves out. And that looks, actually, if you imagine sort of rolling it up, that looks surprisingly like my esophagus picture. So there's a theory of what happens in cancer. There's a theory, I apologize for this being in French, but it comes from a discussion of French Canadians. But it's clear enough even without the words. There's a multi-hit theory of cancer. Cells in general have a lot of fail-safes, so a single mutation is unlikely to result in a cancer. If it did, frankly, our 70-plus year lifespan would be unachievable. So a single mutation occurs. It doesn't lead to cancer by itself, but the theory goes it has some growth advantage. So it grows up into a large clump of cells all carrying that mutation. And that's now an attractive, large target, many cells that could have a second mutation, still not cancer, but giving some type of advantage. Those grow up, and maybe 3, 4, 5 mutations down the row, you've finally disabled so many cellular safeguards that cancer can actually arise. The thing about this model is it pretty much requires each step to have some kind of selective advantage. That's what selection is saying in this graph. If a cell has a mutation that could eventually lead to cancer, but that cell itself has no growth advantage, it's never likely to have more than a few offspring. And that's a pretty small target for the needed second and third and fourth mutation to happen in. So this model has a number of predictions, which Barrett's esophagus does not fit very well. So this sort of multi-hit theory would say that the cancers are going to come out where the mutagens are. We're pretty sure that the mutagens are coming from the stomach and are found mostly at the bottom of the esophagus. But the cancers that arise in Barrett's esophagus, while they can arise anywhere in the Barrett segment, they're actually the most frequent around the middle, not at the bottom. The multi-hit theory also predicts cancers arising gradually over time. So your risk gets higher and higher as you get older and older. But what we see with Barrett's esophagus is that either you get cancer within a few years of diagnosis or you probably never will. That doesn't sound very much like the prediction of the multi-hit model. And finally, it predicts that when we look at the esophagus of someone who did develop cancer, we should see fields of similar cells. Because we're saying that the way the spread happens is that one mutation occurs and its descendants become a big group of cells, and then another mutation happens and its descendants become a big group of cells. But when I look at actual genotypes from cells in Barrett's esophagus and I draw relationship trees of them, that's not what I see. I see all kinds of different mutations. I don't see big sweeps of one mutation covering the whole esophagus. So something is not fitting very well. I'm going to go a little fast because I'm running a bit over time. But this is just where the cancer is found. This is how many samples we had from each level of the esophagus in patients where we did detect cancer. The cancers actually concentrate in the middle. There's definitely not the spike at the bottom that I would expect if the mutagen is really coming from the bottom. So there's an alternative theory for what's going on here. And I want to emphasize this is a theory for Barrett's esophagus and cancer, not for cancers in general. This is an unusual situation. But my theory is this, that the die is actually cast very early. In most people with the Barrett's esophagus, damage to the esophagus triggers the growth of the Barrett segment, and it grows up through, and you're lucky. A bad mutation does not happen during that time. Once Barrett's esophagus is established, it does its job. It prevents mutations that occur subsequently from spreading. So your risk of cancer is very low. Barrett's is actually possibly saving your life here because that crypt structure is really robust in terms of stopping mutations from spreading. In a few people with Barrett's esophagus, they unfortunately hit the jackpot during the initial spread. A bad mutation arose, and just like a single individual in French Canada, it had descendants in this big sector all the way up through the esophagus. Now there is a big target for the second or third mutation that's needed to get to cancer. So this says that by the time we first see a patient with Barrett's esophagus in the clinic, it may be that it's already fairly well determined whether they will get cancer or not get cancer, if only we could see how to read that information out of the genotype of the cells that are already in their throat. We thought that we were doing an ongoing surveillance, waiting for bad mutations to rise to tell us they were going toward cancer. But it may be we should just be asking, what's there now? And if it looks okay, maybe we can leave these poor folk alone and not stick a tube down their esophagus every 6 months for the rest of the 30, 40, 50 years they're going to live, which, frankly, they don't enjoy. So this would explain several of the mysteries. Cancers arise toward the middle because we know that gene surfing pushes mutations in the direction of growth. A mutation will arise, say, at the base of the esophagus, but it's being pushed by the growth thing, so the center of its distribution will be higher up. Cancers arise quickly or not at all because the decision cancer, no cancer is actually made during the initial spread of Barrett's esophagus. And we don't see big selective sweeps during surveillance. We don't see mutations spreading over the 20 years that we've watched the patient because they're not spreading during that period. They're not strongly advantageous. They've been laid down during the growth phase, and now they're either going to sit there and incubate a cancer or they're not. All right. I'm going to skip through. So the implication then is that we could do a lot more to help these people by asking what mutations are present now, rather than by focusing all our efforts on ongoing surveillance. We may be able to drop people out of the surveillance programs and improve their quality of life, and focus our attention on people where we can see trouble brewing already.